In this presentation, the Eurofins DiscoverX portfolio of reporter assays will be described. These cell-based reporter gene assays have been developed to expand our menu of cell-based assays that allow customers to better understand how their candidate therapeutic molecules affect specific signaling pathways. Signaling pathways are a complex series of events that affect a cellular function, usually by changes at the transcriptional level. Some examples of cellular behavior mediated by signaling pathways are cell survival or cell apoptosis, increased or decreased cellular proliferation, or cytokine production by the cells. Shown on the right is one example, the signaling pathway for the NFkB transcription factor, which is labeled as P50 in this diagram. Signaling pathway initiation typically occurs at the plasma membrane with receptor activation and proceeds through a series of cellular events, ultimately resulting in changes in transcription in the nucleus that in turn modify a cellular function. Drug developers have generated therapeutics that can target different proteins or signaling events at any point along a signaling pathway. These drugs are designed to alter specific cellular functions. Reporter pathway assays measure effects of therapeutics on an event near the endpoint of a signaling pathway and therefore can monitor effects of test compounds that affect steps almost anywhere along a signaling pathway. Reporter genes have long been used to identify important transcription factors and regulatory transcriptional elements, as well as being used to measure signaling pathway activity. A reporter gene is comprised of a transcriptional response element that normally drives expression of a target gene in the pathway, but has been engineered in this case to control expression of a reporter protein. When pathway signaling is activated, a change in reporter gene transcription and translation occurs. These effects can be measured by monitoring reporter protein levels or reporter protein activity. Some commonly used proteins for reporter genes are luciferase or fluorescent proteins, since they are readily and easily measured. Thus, the abundance of a reporter protein represents a direct measurement of signaling pathway activity. Effects of therapeutics modulating pathway signaling can therefore be evaluated by measuring changes in reporter protein expression. In the following slides and for the rest of my presentation, I will describe how we have developed reporter gene assays using the well-established Eurofins DiscoverX enzyme fragment complementation technology. So shown on this slide is our enzyme fragment complementation technology, or EFC for short. So this system consists of a split beta-galactosidase enzyme, which has been divided into two fragments, a smaller ED or enzyme donor fragment, and the bulk of the beta-galactosidase enzyme, or the enzyme acceptor or EA fragment. Individually, each fragment has no enzymatic activity, but when mixed together or brought into proximity, the two enzyme fragments can complement to form a functional beta-gal enzyme whose activity is readily measured by the addition of luminescent substrate. For our reporter gene assays, stable cell lines have been generated that contain a reporter gene construct consisting of a transcriptional response element controlling expression of a reporter protein tagged with the smaller beta-gal enzyme fragment. This smaller beta-gal enzyme fragment is labeled as EPL or enhanced pro-label, which we use interchangeably to describe the ED enzyme fragment shown in the diagram on the left. EPL-tagged reporter protein expression is a function of signaling pathway activity and is measured by lysing cells in the presence of the complementary EA enzyme fragment and the enzyme substrate to detect enzyme activity. The advantages of using the EFC system is that we've used this for many of our cell-based assays, and it's a very simple homogeneous mix and read assay format. This assay can be used for high throughput screening in many different microplate formats. First two examples of our pathway reporter assay utilize a reporter gene for the NFkB pathway that I showed earlier in the introduction. Assays with this reporter gene have been developed in two different cell backgrounds, the U2S cell background shown on the left graph and the HEC293 assay shown in the right graph. And these two assays use different activators for this pathway. In the case of the U2S, the CD40L it is used as a ligand, and on the right for the HEC293's TNFα. These ligands are used to activate receptors endogenously expressed by each cell type. TNFα can actually activate the reporter gene in both assays, but data for the HEC293 background is the only one shown on this slide, and that's the data shown on the right. However, only the U2S cell line responds well to the CD40 ligand as shown in the graph on the left. This difference in the response to CD40L in the two assays is likely a consequence of reporter expression profiles for each background, where the HEC293 exhibits little to no CD40 receptor expression. Thus, reporter genes can be tailored for specific activators of a particular signaling pathway by choice of cell background used for the assay. Alternatively, cells can be engineered to express the target receptor of interest to activate the pathway, and this is what we did to develop an assay for rank using the U2S NFkB reporter assay. So on this slide is shown the data with the rank NFkB pathway reporter assay, which was created by rank gene transfer into the U2S NFkB pathway reporter cells. And this was used to generate a sensitive assay for rank activation by rank L with an EC50 value in the nanogram per mil range, and this data is shown on the left. In the graph on the right, results from testing with a therapeutic rank L inhibitor Prolia, which is currently used to treat osteoporosis, demonstrates a potent inhibition of rank activation in response to rank L. In this slide are results from another of our reporter gene assays that use an endogenous receptor expressed by the cell background used to generate the assay. And this is the HepG2 STAT3 reporter assay. In this assay, IL-6 is used to activate the STAT3 pathway in HepG2 cells, and this data is shown in the graph on the left. Potency of IL-6 is in the sub-nanogram per mil range, and so it is very good at detecting activity with good potency. Recently, the contribution of IL-6 receptor pathway signaling to outcomes of COVID-19 patients was documented. Elevated IL-6 levels have been observed in COVID-19 patients. Tocilizumab, an immunosuppressive drug currently used to treat rheumatoid arthritis, has recently been approved for phase 3 clinical trials on COVID-19 patients with pneumonia. Data on the right shows results using the HepG2 STAT3 assay, testing tocilizumab inhibition of IL-6 activation of the STAT3 reporter. These results with tocilizumab demonstrate the potential utility of this reporter gene assay for the development of new therapeutics. So another reporter gene assay that we've developed is presented here and measures T cell activation occurring through the T cell receptor. This assay provides a much easier and quicker alternative to measuring T cell activation using proliferation or cytokine secretion assays that have been used previously. Here in the graph on the left a potent activating CD3 antibody was used to activate the JRCat NFAT reporter genes. And this was done by pre-coding the wells with increasing concentrations of the CD3 antibody on the assay plates, and then plating the NFAT reporter cells into the wells coated with antibody. With increasing concentrations of antibody, increases in T cell activation and NFAT reporter gene expression are observed. The data on the right demonstrates the assay in antagonist mode, where a small molecule inhibitor of protein kinase C was used to inhibit the signaling pathway occurring downstream of T cell receptor activation, and thus inhibiting NFAT reporter gene expression. This reporter gene assay is another example of an assay that can be used as a building block to further modify the assay so that other cellular signaling inputs affecting the pathway, in this case T cell activation, can be assessed. The immuno-oncology checkpoint receptors have been targets of intense drug development due to their therapeutic value in stimulating the immune system to combat cancer. Immune checkpoint receptor activation results in inhibition of T cell activation and suppresses the immune response. Activity of checkpoint receptors can therefore be measured with the NFAT reporter. Therapeutic inhibitors of checkpoint receptors result in greater T cell activation. One such checkpoint receptor that has been of intense study has been the PD-1 checkpoint receptor. Here we demonstrate how the JRCat NFAT reporter assay cells, which measure T cell activation, were further modified to generate an assay to measure the effects of PD-1 checkpoint receptor signaling on T cell activation. In this case, pathway reporter assay cells were used in a co-culture system with cells overexpressing a T cell receptor activator and used to activate the NFAT reporter gene instead of the CD3 receptor antibody that was used in and shown on the previous slide. When JRCat reporter assay cells and increasing concentration of the U2S activator cells are cultured together, the result is a large signal increase due to T cell receptor and T cell activation. When PD-1 is expressed in the reporter gene cells, this is shown in the blue curve, there is a reduction in the amount of T cell activation when in these co-cultures. When PD-L1 is added to the is co-expressed with the T cell receptor activator cells and co-cultured with the PD-1 NFAT cells, an additional attenuation of the T cell activation is observed, and this is shown in the red with the red curve. So T cell activation is reduced due to PD-1 activation by the PD-L1 ligand, and this PD-1 activation results in inhibition of the T cell activation. Thus, by adding the PD-1 receptor to the reporter cells and the PD-L1 ligand to the corresponding U2S TCR activator cells, we get a reduction in the amount of T cell activation. So this system can be used to screen for therapeutic molecules that can release PD-1 inhibition on T cell activation. The specifics of how this assay works is shown on the next slide. So this schematic again illustrates how the PD-1 NFAT reporter assay works in this co-culture system. So again, this assay consists of two cells that are co-cultured together, where one cell line is used to present the ligand for PD-1 as well as the activator for the signaling pathway So it consists of U2S co-expressing the T cell receptor activator and the PD-1 ligand, or PDL1. And these cells are cultured together with the JERCAT NFAT reporter cells co-expressing PD-1 to now make them responsive to PDL1. When the two cell lines are cultured together, some T cell activation is measured, but as shown on the previous slide, it is reduced compared to the co-culture of cells without PD-1 and PDL1 co-expressed. Thus, when an inhibitor of PD-1 or PDL1 is added to the culture, as shown on the right, so in this case we're adding a anti-PD-1 antibody, PD-1 activation occurs. PD-1 activation and its inhibition of T cell activation is blocked, and greater levels of T cell activation are observed as PD-1 inhibition is released. This PD-1 NFAT reporter assay thus measures the cumulative effects of negative and positive pathway inputs from PD-1 and T cell receptor activation respectively by monitoring transcriptional effects well downstream of both events. The results from this PD-1 NFAT reporter assay contrasts another pathway PD-1 pathway signaling assay we offer in our PathHunter assay portfolio, which is not based on a reporter gene readout. This schematic illustrates where along the PD-1 signaling pathway the two different PathHunter PD-1 assays measure PD-1 activity. Both assays can be used to measure PD-1 signaling activation and inhibition, but as we have described in previous slides, the pathway reporter gene assay measures a later event in the sequence of signaling, PD-1 signaling, and this event is boxed in purple on the right, while the PathHunter JERCAT PD-1 signaling assay monitors an event occurring earlier in the sequence. Again, this event is boxed in blue and occurs soon after PD-1 receptor activation by ligand binding, but prior to PD-1 effects on activating signals mediated through the T cell receptor. Both assays provide results that can be used to evaluate therapeutics designed to activate the immune system by blocking PD-1 inhibition, but each provides slightly different information. The signaling assay monitors a more proximal event occurring at the plasma membrane with PD-1 receptor activation that is independent of T cell receptor activation, while the PD-1 reporter gene assay measures the cumulative effects of TCR activation and PD-1 activation or inhibition that results in downstream transcriptional changes. Additional details of how the PD-1 signaling assay works is presented in the next slide. Like the PD-1 NFAT reporter assay, this PD-1 signaling assay also uses a co-culture format where ligand cells are used to activate PD-1 in the signaling cell line. JERCAT cells expressing full-length PD-1 receptor engineered with the small beta-gal enzyme donor fragment which is fused to its C terminus. This is co-expressed with the SHP1 or SHP2 phosphatase fusion protein with the complementary beta-gal enzyme fragment fused. Ligand engagement by co-culturing with ligand-presenting cells results in tyrosine phosphorylation of the checkpoint receptor PK fusion protein in the cytosolic item motif of PD-1, leading to the recruitment of the SHP1 EA fusion protein through the SH2 phosphotyrosine binding domain of SHP1 or SHP2. Complementation of the two enzyme fragments occurs and results in an active beta-gal enzyme. This active enzyme hydrolyzes substrate to create a chemiluminescent signal, which is a measure of receptor activity. When an anti-receptor or anti-ligand antibody is added to the culture, both ligand receptor engagement is disrupted and no SHP1 fusion protein recruitment or enzyme complementation occurs. Because the assay measures PD-1 receptor activation prior to its effects, on T cell activation activity, the readout from this assay is unaffected by T cell activation. So presented on this slide is the data from both PD-1 assays using a PD-1 antibody to block PD-1 signaling and its inhibition of T cell activation are shown in this slide. Inhibition of PD-1 activity mediated by co-culture with PD-L1 ligand cells in the signaling assay, shown in the left graph, results in a loss of signal as PD-1 receptor activation is inhibited and SHP1 or SHP2 is no longer recruited to the receptor with the higher concentrations of antibody used to block. In contrast, higher PD-1 antibody concentrations in the reporter assay format blocks PD-1 attenuation of T cell activation by the TCR activator, and a gain of signal is observed due to greater T cell activation occurring with the release of PD-1 inhibition of T cell activation. Both assays measure PD-1 signaling with similar potencies in the 100 nanogram per mil range, despite the fact that they are measuring two different events in the PD-1 signaling pathway. The PD-1 signaling activity is a more direct measurement of PD-1 receptor activation that is independent of TCR activation, whereas the PD-1 NFAT reporter assay is measuring the sum effects of blocking PD-1 activity and its effects on T cell activation through the TCR on Jurkat cells. Thus, our two different PD-1 assays provide an example of how two different assay formats for the same signaling pathway enable a customer to extract different kinds of information about their therapeutic molecules, depending on which assay is used. In this slide, I've more generally tried to illustrate the differences between reporter gene and non-reporter gene assay formats for the same pathway. Reporter assays measure a very distal event in a signaling pathway. They measure transcription and therefore represent the cumulative effect of signaling from multiple inputs modulating a response. So in the example of PD-1, it's measuring both TCR activation and PD-1 inhibition. Since transcriptional changes from pathway signaling are very downstream, it represents is much more representative of the ultimate cellular response changes. Results with the reporter gene represent the cumulative effects from a signaling pathway on cellular responses and can monitor different inputs affecting the pathway as well as effects on events almost anywhere along the pathway. In contrast, non-reporter gene signaling assays typically measure a specific event in a pathway upstream of transcriptional effects and can more directly address the MOA of a drug. In the case of PD-1, this event is proximal to PD-1 receptor activation, but prior to PD-1 effects on signaling from the T cell receptor activation. Both types of assays, as illustrated by our complementary PD-1 signaling assay and NFAT reporter gene format, yield results with robust signal to background ratios and demonstrate good potency with therapeutic molecules. Both assays have short assay times and utilize our EFC system to make them simple and easy to run. Our PD-1 signaling assay is currently already available in bioassay format for QA applications. Thus, the two PathHunter PD-1 assay formats provide customers options for how they would like to test their therapeutics and in order to generate results that more closely meet their goals or needs. Finally listed here are the current list of PathHunter pathway reporter cell lines we have available and the link on our website to obtain more information about our reporter assays. I'd also like to point out that we offer our custom assay development service to develop new reporter gene assays or modify currently existing reporter gene assays to fit a customer's specific target or need. Cell-based assays are valuable tools throughout the drug discovery process, but internal assay development can be challenging, expensive, and time-consuming. Our services take advantage of both our extensive cell-based assay development and cell engineering expertise. The email address for CAD inquiries are located below, and I encourage you to contact our CAD assay development team. In this last slide, I would like to summarize my talk by reviewing the advantages of our PathHunter pathway reporter assays. These assays take advantage of our well-established enzyme fragment complementation system and make the assays extremely quick and easy to use, and are well suited for high throughput screening. The assays yield rapid responses, especially compared to results from phenotypic cell assays. I've shown validation assay with our reporter genes cell lines that demonstrate the specificity and sensitivity of the assays, as well as how well these assays can be used to test both small molecule inhibitors and biologics. The addition of reporter gene assays to our already expansive portfolio of pathway assays with non-reporter gene formats provides an alternative readout at the distal end of a signaling pathway. I have presented data from both our PD-1 NFAT reporter assay and PD-1 signaling assay as an example of the two assays monitoring distinct events in the same signaling pathway, either a distal event like reporter gene expression or a more receptor proximal step that together can provide complementary results for drug discovery. And finally, these reporter gene assays not only provide an off-the-shelf tool for evaluating drugs, but also provide a building block for development of additional pathway assays. I'd like to thank you for your time and attention today, and invite you to visit our website to learn more about our cell-based pathway reporter gene assays.